This video mainly introduces the collection and detection process of intestinal lavage fluid, fecal swab and whole blood samples of InCycle automatic nucleic acid analysis system. 1. Checking and preparation. Open the kit and check if the components are complete according to the instruction. The kit contains 3 different tubes. Tube 1 and Tube 2 are 2 kinds of sample preservation solution, and Tube 3 is a sample treatment tube. In addition, it also contains a centrifuge tube and 0.5 ml EDTA anticoagulant tube. Before conducting the test, mix the magnetic beads well by shaking them gently. Tear open the packaging bags of reagent to check appearance of lyophilized PCR reagent. Avoid using the moisture-absorbed reagents as shown in the picture. Next, gently flick the lyophilized PCR reagent to ensure it is located at the bottom of the tube. Check for the bubbles in the PK liquid. If present, gently flick the reagent from the bottom of the tube to eliminate the bubbles. Then, gently flick the PK liquid reagent to ensure it is positioned at the bottom of the tube. The reagent should be completely at the bottom of the tube. 2. Inserting reagents. Align tube 1 with the reagent tube slot. Insert it into the corresponding position of the cartridge and place it as shown in the figure. You can lift the reagent tube up with your fingers. A firmly fixed reagent tube indicates successful tube installation. Then tear off the cartridge sealing film and insert the PK into the corresponding position. 3. Sampling and sample processing. Fecal swab, intestinal lavage fluid, and whole blood are the applicable types of samples. Now the pre-treatment method of intestinal lavage fluid is introduced firstly. After collecting the intestinal lavage fluid, use a 0.2 ml disposable dropper to transfer 0.2 ml of fluid into tube 3. Tighten the cap securely and centrifuge it at 12,000 rpm for 2 minutes. Once done, carefully discard the supernatant, retaining the bottom precipitate. Next, replace the dropper and add 0.6 ml of the pre-treatment solution from tube 2 into tube 3. Tighten the cap and place tube 3 into the vortex mixer for 3 minutes, ensuring thorough mixing of the precipitate with the pre-treatment solution. After vortexing, centrifuge tube 3 at 12,000 rpm for 2 minutes to obtain the processed sample. Then use a new disposable dropper to collect the supernatant up to 200 micron mark, which can be used as a sample for testing. Now, let's discuss the pre-treatment method for fecal swab samples. Gently dip the head of the disposable swab into the fecal sample until it covers approximately 1 second to 2/3 of the head surface. Thoroughly mix the swab in tube 1. Using a disposable dropper, transfer 0.2 micron of the fecal mixture from tube 1 to tube 3. Tighten the cap and centrifuge tube 3 at 12,000 rpm for 2 minutes. Carefully remove tube 3 and discard the supernatant, retaining the precipitate at the bottom. Carefully replace the disposable dropper. Add 0.6 ml of the preservation solution of tube 2 to tube 3. Afterward, vortex the mixture in tube 3 for 3 minutes. During vortexing, mix the tube intermittently to ensure proper dispersion of the precipitate in the preservation solution. Following vortexing, centrifuge tube 3 at 12,000 rpm for 2 minutes to obtain the processed sample. Replace the dropper with the new one and collect the supernatant up to 200 micron mark to use as the sample for testing. Moving on to the pre-treatment method for whole blood samples. For specific preservation requirements of blood samples, please refer to the whole blood samples collection and detection process video. Using a disposable dropper, add 0.6 ml of the preservation solution from tube 2 into a 1.5 ml centrifuge tube. Then replace the dropper and add 0.4 ml of whole blood to the same centrifuge tube. Mix it thoroughly by inverting the tube for 2 to 3 minutes, or by vertexing it for 10 seconds. This constitutes the processed sample. Note: processed samples should be tested as soon as possible. Replace the dropper again with a new one and collect the mixture up to 200 microliter mark to use as the sample for testing. 4. Adding sample and testing. Add 200 microliter of the sample supernatant or mixture dropwise into the chamber of the centrifuge sample compartment, and click the red exit button to exit the sample drawer. Place the cartridge in the analyzer in the order of left sample compartment, right PCR. Hold the buckle to the right, click the red exit button to enter the sample drawer, and click Test, followed by Next. Before testing, swipe the RFID card in the card swiping area on the side of the analyzer. When swiping the card, keep it stationary until the beep ends. After successfully enter the information, confirm the sample type and other basic details. Then follow the analyzer prompts and click the button in sequence to start the test and proceed accordingly. 5. Threshold adjustment and interpreting the results. After the test, please confirm the amplification curve before outputting the report. If you need to adjust the threshold, please refer to the threshold adjustment operation video. After confirming the results, click Audit OK. Subsequently, you can view and print the test report in the InHub software. Note: only approved results can be viewed and printed. 6. Cartridge handling. Click the red exit button to exit the sample drawer, and affix a seal sticker, yellow, to the surface of the cartridge. Then, put the buckle to the left to remove the cartridge. Used cartridge should be handled in accordance with the requirements of the local regulations of pollutant treatment processing. G-Links. Everything is for life.